So in this week's lecture, we're going to cover antigen presentation to T cells. And it's the beginning of a multi-chapter series on T cells. We previously covered migration of all leukocytes, including T cells and B cells. And now we're going to start talking about the antigen presentation. Activation comes in the next chapter, and then the differentiation of the two main subtypes of the effector T cells, the CD4 plus and CD8 plus cells. So it's a good indication of the intricacies of the adaptive immune system, how we have a multi-chapter series just on these cells, and we do something very similar with B cells later on. But let's get into it. Now, the purpose of T cells is really twofold. The first is that they help eradicate infections of intracellular microbes and also cancer cells as well. The second principal function is that they help activate other cells like macrophages and phagocytes, and they're also vitally important for the development of B cells. And both of these help function to eradicate microbes of the extracellular space. And I like to say that a very quick summary of this is that killer T cells of the CD8 plus variety help eradicate cells directly in the intracellular space. And then the CD4 plus cells, the helper T cells, help other cells accomplish getting rid of microbes in the extracellular space. So a couple key things that make T cells particularly special, and some of these we learned in previous chapters, like in the migration of lymphocytes, is that the antigens for T cells are captured at the site of entry through antigen-presenting cells, the most common one or most powerful one being dendritic cells. And then they concentrate these antigens in the secondary lymphoid organs, which act as a great way for T cells to circulate through. And that's a point I want to stress here, is that T cells are in this constant state of migration and recirculation, strictly in the hopes of finding their antigen. And in fact, 99 point and then 9 times, many times repeating, of T cells that ever get created by your body never find their antigens, which you could argue is a good thing. One of the reasons why antigen-presenting cells are so important for T cells is that it's impossible for T cells, or B cells for that matter, specific to any antigen, to constantly patrol in all sites of tissues where you might get an infection in order to recognize their antigen. So the use of antigen-presenting cells to present to these lymphocytes is really a means of necessity in order to present their antigens. Now, the importance of antigen-presenting cells is also highlighted by the fact that in T cells in particular, that T cells only respond to cell-associated antigens. They have to have antigens presented to them. They cannot recognize free soluble ones that are not associated with any cell. So T cells can't even recognize a microbe directly. This is a big contrast between B cells, which can do so. The antibody receptors on B cells can recognize solubilized antigens. T cells have to get it from another cell through an antigen-presenting cell. And one of the key features of these antigen-presenting cells is that they do their antigen presentations through compounds of the cell surface known as major histocompatibility complexes, MHCs. So those are the clusters that are responsible for presenting antigens to T cells. We'll learn more in detail later why this is so important, but basically it's to help present antigens to the right subtype of T cells. Whether you have an intracellular microbe, you want these antigen-presenting cells to call upon the CD8 plus variety. If you have extracellular microbes and you need help from T cells, you need to present the cells to the CD4 plus variety. This is a concept known as MHC restriction, because different MHC subclasses only present to certain types of T lymphocytes. Another big difference between T cells and B cells is that T cells can only recognize short peptides. B lymphocytes, on the other hand, can not only recognize large peptides, but any other type of molecule as well. Nucleic acids, lipids, carbohydrates, small molecules, large molecules. So B cells have a much larger repertoire of antigens that they can recognize. And the only really natural way for T cells to recognize molecules other than peptides are molecules that can bind to peptides themselves. This is actually common with metals. Some people that have metal allergies, it's likely due to T cells recognizing chelating abilities of some metals to bind to peptides, and thus get presented to T cells as an antigen. So now I'd like to explain a little bit about the history of our understanding of antigen presentation to T cells. So in early studies, Scientists would take an antigen that was labeled, and they knew prior to this that it had some known T cell specificity. Okay, they knew at this time that lymphocytes were associated with recognizing antigens in the body, and that the adaptive immune system was a special arm of the immune system. So they took an antigen that was labeled known T cell specificity and gave it to mice, and then they isolated different kinds of cells to see where that antigen ended up. And to their surprise, it was bound to non-lymphoid cells as opposed to T lymphocytes. So they thought, okay, well, why is it that T lymphocytes are reacting so much to this antigen, yet this antigen is not associated really in any way with these T lymphocytes? So in later studies, they would purify and isolate just the CD4 plus variety of T lymphocytes, and they try and pair it with that same antigen. And nothing would happen. And they thought, again, okay, we know T cells are involved with the recognition of this antigen. Why is nothing happening here? So in the second set of experiments, they did the same thing, this time adding a mixture of non-lymphoid cells like dendritic cells and macrophages, and then all the magic would happen. Again, these non-lymphoid cells would associate with the antigen, and then they would see the activation process of the CD4 plus cells. And these kind of experiments led to the concept that there's a critical step in induction of the T cell response through antigen presenting cells. And now that we're getting into antigen presenting cells a bit more in detail, I'd like to cover a few basic properties of them. The first is that there are actually many different types of cells that can serve as antigen presenting cells, and some of them are more useful at various stages of activation. And I like to preface that by saying that I think up until now, especially when I was first going over this material, I understood activation as sort of a binary state. You're either naive or a cell was activated and ready to go. And that's only really partially true in that once a lymphocyte has been presented with an antigen, it's no longer naive. But there's different stages of activation, both during the differentiation process into the exact subtype of T lymphocyte or even B lymphocyte that might be necessary at a given moment. And also, effector cells need to be reminded to stay activated and to carry out their functions. So antigen-presenting cells are needed all throughout the process of being activated. Otherwise, like in the case of an infection dying out and antigens no longer being present, the T cells are no longer necessary. So now going back to APCs, dendritic cells are really the rock star of the APC group. They're the most effective APC for activating naive T cells. We learned previously that they have the most diverse array of toll-like receptors, so they have all the necessary equipment to help activate dendritic cells, and they also migrate around the most as well. So they respond to a variety of chemokines, more so than other non-lymphoid cells. On the other hand, macrophages and B lymphocytes, while they do serve as APCs, are really only needed for previously activated CD4 plus cells. So usually in the timeline, it's dendritic cells first, and then other APCs, or dendritic cells including, to serve for the already previously activated cells. In fact, B cells and B lymphocytes are only useful to serve as APCs for previously activated CD4 plus variety T cells. And that's because they only have the MHC, the major histocompatibility complex, that can be recognized by CD4 plus cells, and not other subvarieties of T cells. Dendritic cells, on the other hand, as we're going to learn, are able to express both, and thus not only activate naive cells in the various subtypes, but then also continue activating both CD4 plus and CD8 plus cells down the road. And part of the activation and continual activation of T cells is that APCs need co-stimulators along with their antigen MHC complexes to help T cells along. So we learned previously this is part of the signal 1, signal 2. Now those signal 2 systems are usually membrane-bound mechanisms of further stimulating T cells. There's also cytokines that APCs give out to help further activate or differentiate T cells. Now, in order for APCs to really carry out these functions, they have to recognize that there's something foreign, something dangerous in the body. So all this antigen presentation is enhanced by exposure to microbial products. For this reason, whenever we develop vaccines, in addition to the purified antigen, you also need to inject what are known as adjuvants. These are sort of co-stimulators or even portions of microbes and products other than the antigen to help stimulate APCs into activating T lymphocytes. And then lastly, as sort of a positive feedback mechanisms, lymphocytes themselves give signals to APCs to enhance their antigen-presenting functions. Now that we get to the rock star of the APC world, I'm going to play an animation from the book. Enjoy.
Immature dendritic cells in the epithelium capture microbial antigens, are activated, and leave the epithelium. The dendritic cells migrate to draining lymph nodes, being attracted there by chemokines produced in the lymphatics and nodes. Following activation, the dendritic cells mature and begin to express high levels of major histocompatibility complex molecules and co-stimulators, which function to stimulate T cells.
Just fascinating. Absolutely fascinating. Now, the most common way for pathogens or microbes to enter a host are through epithelial membranes, like in the respiratory tract or skin or mucosal layers. And this is where they be concentrated in the lymph nodes via classical dendritic cells that are present and resident in these tissues as just sentry cells. Another common mode of entry would be through bloodborne agents. And these would either concentrate in the spleen, where resident macrophages there would then bring them to the lymphatic system, or actually be captured directly by circulating plasmacytoid dendritic cells and then taken to the lymphatic system that way. Now, as dendritic cells process antigens to present them to lymphocytes, usually through typical phagocytic means, they independently also get activated by microbial products like LPS, flagellin, things that are recognized by toll-like receptors or other pathogen-associated molecular patterns. And I want to stress that this is totally independent of the antigens that they process. Very often, the dendritic cells don't care at all about the antigens. They're made to process them and then present them to lymphocytes, but that's not what I'll say activates them. Okay, they need to recognize something foreign through like their toll-like receptors, their nod-like receptors, and other pathogen recognition markers. But that's separate from the antigens that they present to T cells. So as they recognize these pathogen-associated patterns, that helps them mature into more potent APCs. They lose their adhesiveness to the epithelia. So now I want you thinking both selectins, like P and E, and the ligands for those, and then the integrins as well. They start expressing CCR7, which makes them want to flow into the lymph nodes. And they also express high amounts of MHC, these major histocompatibility complex, and co-stimulators that, as we learned in the video, are necessary presenting the T cells. Now, even if there aren't these pathogen-associated molecular patterns, like there would be in an absence of an infection or inflammation, dendritic cells still have a lot of use because they can still bind to antigens and present them to lymphocytes for the purpose of reporting self-antigens or generating regulatory T cells, because they process all types of antigens, whether they're self or host-derived or foreign-based. Now, there's 4 key traits that make dendritic cells more advantageous in terms of antigen presentation than their other APC counterparts like B cells or macrophages. The first is that dendritic cells are strategically located at common sites of microbial entry, right? So sites like the epithelia, in tissues that may be colonized by microbes. Second is that they express receptors that enable them to capture and respond to microbes. As I mentioned before, they have the highest amount and variety of toll-like receptors, nod-like receptors, cytosol-like receptors, any pathogen molecular patterns. Dendritic cells express them more and in more varieties than other APCs. Third, dendritic cells have the capability to migrate from epithelial tissues via lymphatics to common sites of T cells. So now again, I want you thinking Cristiano Ronaldo, number 7, Creedence Clearwater Revival. That means CCR7. That's how I always remember it. Might be dumb, but it works. And lastly, they express high amounts of antigen MHC complexes. We'll learn later that they express both subtypes of MHCs. You don't need to know that now, but I want to throw it out there. Co-stimulators and cytokines necessary to activate naive T cells. This really makes them the most advantageous to activating naive T cells as opposed to activating or continually activating ones that have already been activated. Geez, can I say activated even more? Come on. Now, to highlight the importance of dendritic cells over other antigen-presenting cells, I'm going to squeeze all other APCs on a single slide. So the first one is macrophages. So macrophages, of course, have the ability to phagocytose microbes and other pathogens. They then have the ability to present antigens of those microbes to T cells, like CD4 plus cells, and these subtypes of T cells will then instruct the macrophages to kill their phagocytosed cargo. B cells also have the ability to internalize protein antigens and present them to CD4 plus cells. And this is vitally important for the development of more specialized and more effective forms of antibodies. It's a process known as T cell-dependent antibody production. And lastly, in a more generalized form, is that all nucleated cells have the ability to present to the CD8 plus variety, the cytotoxic T lymphocytes, which is important because all cells are capable of getting infected by viruses, and all cells are capable of forming cancer. And CD8 plus variety of T cells are the variety to kill internalized infected cells directly or cancerous cells directly. Now, because all cells are capable of doing this, we don't typically call all cells antigen-presenting cells. And what we sometimes do is even call dendritic cells, macrophages, and B cells professional APCs to help differentiate this. So now, before discussing the functions of the major histocompatibility complex, I'd like to talk a little bit about the history of its discovery. Now, it had been known for a long time that tissue transplantation would be rejected by non-identical individuals, but could be successful through identical twins, right? So it was known that this was somehow based on genetics, but this was still poorly understood. Therefore, a group of scientists started inbreeding mice. Now, inbreeding involves the continuous breeding of siblings through many generations until they are homozygous, which means that the alleles from each parent are identical, so each inbred mice has 2 copies of the same alleles. What this guarantees is that each mouse within the same inbred strain is genetically identical. Now, they could do this with different sets of mice so that you can have different inbred strains that would then be allogenic, which means while the inbred mice might be genetically identical to each other, they would not be identical to different strains of inbred mice. So what they would do is that they would attempt grafts between these different strains, and they would inbreed them specifically so that the difference between the 2 different strains of mice would only differ in a single gene. And because these 2 different strains only differed in a single gene, they would most commonly accept the tissue transplantation, except for in one particular case where the rejection occurred, and thus the scientists could conclude that that gene was somehow responsible for tissue transplantation rejection. And for this region, once they isolated that gene, they called that region the major histocompatibility complex. Now, MHC in this case refers to the gene that they later found out encodes the complex that sits on the membrane. So they confusingly have the same exact name, but that's just because the same complex is the gene that forms the membrane-bound protein complex of the same name. And this is actually the source of the name histocompatibility complex. Histo, I think, meaning tissue, like when you think of histology, the study of tissue samples, and then, of course, compatibility. Now, at this point, scientists knew that first, the success of tissue transplantation was genetically based, and that the source of this genetic basis was through MHCs. But this is actually quite confusing. So they knew this for a few decades, yet no one could really explain why vertebrates would have evolved to have this feature. It's not like our evolutionary ancestors were attempting to do lots of tissue transplant or grafts. So why the body would need to evolve a mechanism to reject this and target it was beyond any explanation of any scientists. So to investigate this further, there's a famous study that continued on this line of using different inbred mice where in one mouse strain, we'll call it strain A, the mouse was infected with a type of lymphocytic virus. And after a period of 7 days, the mouse would have activated T cells. They would produce T cells, cytotoxic killer cells, specific to this virus that could effectively kill and fight off the infection. And then what they would do is co-culture these CTLs with target cells of both that same uninfected strain A and then a different strain B, whose only difference was through the MHC complex. What they found was that the cytotoxic T lymphocytes specific to strain A were, of course, successfully able to fight off the infections of the infected strain A cells, so cells that were infected from the same exact genetically identical strain A. Yet, when co-cultured with infected cells of strain B, ones whose only difference was in the genes that encoded the MHC complex, also again called MHC, was that these CTLs were unable to fight off these infected cells. What scientists could then conclude from this study was that CTLs are not only specific to the antigen that they recognize, in this case both strain A and strain B would have had the same antigen, but to the MHC complex that presents that antigen. The two scientists were Rolf Zinkernagel of Switzerland and Peter Doherty of Australia. And for this famous 1974 study, they would be awarded the 1996 Nobel Prize in Medicine. Now, going back to the expression of major histocompatibility complex, I had alluded to this earlier, this concept of class restriction. And what that means is that there's two different classes of MHC molecules, okay, that are appropriately called class 1 and class 2. And T lymphocytes are class restricted to the types of MHCs that they can recognize, because there's two major subtypes of T lymphocytes, CD8 plus cells and CD4 plus cells. CD8 plus cells can only recognize the class 1 MHC molecule. Now remember, that's expressed on virtually all nucleated cells because all cells can get infected with viruses. All cells are capable of developing cancer or becoming cancerous. CD4 plus cells are a little bit more limited because only these helper T lymphocytes can recognize class 2 MHC molecules, which are really only expressed on dendritic cells, B lymphocytes, macrophages, and then a few other smaller cells that we don't really talk about too much, like thymic epithelial cells. But I want you to remember those top three: dendritic cells, B cells, and macrophages express the class 2 MHC, and only CD4 plus helper T cells can recognize those. Now, the expression of MHC molecules are increased by cytokines produced during the innate and also adaptive immune responses. So, for instance, type 1 interferons secreted during the infection of viruses appropriately help express class 1 MHCs. Again, CD8 plus cells are the primary fighters against viruses. And then class 2 MHCs are stimulated by dendritic cells and macrophages in response to IFN gamma, for instance, produced by natural killer cells. So now let's get into the general structures of major histocompatibility complexes. So on the picture on the right, I'm hoping that there's a few elements that look familiar, such as Ig domains and disulfide bonds, and hopefully you think, hey, there are a lot of similarities between this and antibodies that we covered in the previous chapter. And you're correct. MHCs are part of the Ig superfamily because of these similarities. Now, there are a few things common with both classes of MHCs, such as on the upper portion an extracellular peptide binding cleft, and this is the part that actually holds and binds the peptide to be displayed to T cells, then the Ig-like domains, and then finally a transmembrane cytoplasmic domain. Now, the polymorphic amino acid residues that are genetically coded and thus different from person to person are located in the cleft, so in the top portion of the MHC structure. And again, this is what binds to peptide. Now, despite this genetic variability from person to person, I want to stress that MHCs are not the hypervariable analog to antibodies or antibody receptors on B cells. The analog in that case would be the T cell receptor, and that's what can recognize, again, the 10 to a 7, 10 to a 9, a huge range of different antigens. The MHC structure, while being variable, binds much less selectively in that one MHC individual molecule binds to many different types of peptides and can process and present many different types of antigens and are not specific. Again, that would be the T cell receptor, like the B cell receptor or the membrane-bound antibodies. Now, going back to the structure, the Ig-like domains contain special binding sites or docking areas that help bind to the T cell. And for that reason, they're sometimes called co-receptor. They again act like docks. And they do differ between the two structures. They're either of the CD4 variety and CD8 variety. And this is actually what give T cells their names, because this is what determines what kind of T cell can bind to that particular MHC, whether or not it has a CD4 dock or a CD8 dock. And again, a CD4 plus cell will bind to the CD4, CD8 plus cell will bind to the CD8. And these particular docks are not polymorphic, so they're not genetically encoded by the MHC gene. So now let's discuss the specific structure of class 1 MHCs. So class 1 MHCs consist of two chains. There's the long alpha chain and the short beta chain. As you can tell, the alpha chain is the part that contains the peptide binding cleft, and because that's the part that's polymorphic, is encoded by MHCs, whereas the beta cleft are not. Now, the alpha chain is divided into three segments, and alpha 1 and alpha 2 terminals, which are about 9 amino acids in length, they're what form the peptide binding cleft. And they can only recognize peptides, really short peptides, about 8 and 11 amino acids in length. But if you think this is small, remember that there are about 20 essential amino acids, I think. So 20 to the 11th power makes up a huge amount of different peptides that can form. So you can get really specific here. And then finally, the alpha 3 segment is the portion that's involved with binding to CD8 plus cells, along with a little help from the beta chain and portions of the alpha 2 segment. But the alpha 3 segment is the part that's conserved amongst all class 1 MHCs. So despite any variability in the alpha 1 and alpha 2 segment, alpha 3s for all class 1 MHCs are going to be exactly the same. And they finally extend down into the transmembrane region, which consists of around 25 hydrophobic amino acids. Remember, the lipid bilayers is hydrophobic. And then into the cytosol region, where there's about 30 amino acids, basic amino acid residues that help anchor the MHC. Now, one thing to know about actually all MHC structures, not just class 1, is that the stable assembly and expression of them requires a bound peptide. Okay, so I even think the picture here on the right, especially the cartoony one on the left, is a little misleading because there's no peptide there, and you would never find that on a cell. Okay, it always has to have a bound peptide, and we'll learn just in the last part of this lecture how MHCs are formed inside the cell with the peptide, and that's what helps them get expressed. But this help ensures that only potentially useful MHC molecules are expressed, not ones without any bound peptide. Now moving on to class 2 MHC structures, we're going to have a lot of similarities between the two. But let's start with some of the differences. So what I see is instead of having a long alpha chain and a real short beta chain, you have almost two identical and symmetrical chains, each labeled, or one labeled alpha, one labeled beta. Key difference here is that both alpha and beta chains are MHC encoded. Remember, class 1 only has the alpha chain that's MHC encoded. So in this case, both chains are polymorphic. Yet, the polymorphic residues are really located just in the cleft binding region in the alpha 1 and beta 1 segments. In fact, most of the polymorphism is concentrated on the beta 1 chain. And another key difference between class 1 and class 2 is that the class 2 can recognize amino acid peptides 30 amino acids in length. So this is actually a lot longer than the class 1 structures. Now, just like in class 1 MHC structures, the Ig-like domains, in this case alpha 2 and beta 2, are conserved amongst all class 2 MHC structures. So these are not polymorphic, and these help bind to their respective docking co-receptor on the T cells. Remember, class 2 gets recognized by CD4, and remind again, class 1 gets recognized by CD8. And just like class 1 structures, the stable assembly of all class 2 MHCs require bound peptides. So you would never find a fully intact class 2 MHC without a peptide. They don't exist. So now we're going to get into the actual peptide binding to MHC molecules. So I want to give a brief overview of some of the key characteristics about this process. First, like I stressed before, is that MHCs need to show a broad specificity, okay, and that in contrast to the T cell antigen receptors on the actual T cells, okay? So even though these are genetically encoded, there's not a lot of diversity amongst them within an individual, okay? I think you're capable of producing 6 different kinds of class 1 MHCs. I think anywhere from 8 to a dozen class 2 MHCs. So you don't have a lot of variety within a single individual. And for that reason, a single MHC has to be able to present many different peptides. And again, only a single T cell can recognize one specific peptide MHC combination. Now, each class 1 and class 2 MHC molecule can only bind a single peptide at a time. So only one peptide, one antigen is associated at a given time with an MHC molecule. And this can actually work against us because studies have shown that different peptides and antigens can outcompete MHC molecules whenever they're presenting on a cell, which can work against the presentation of a specific antigen that's necessary to link up with a specific T cell. As I mentioned when we were talking about the structures of these complexes, MHC molecules acquire their peptide cargo during their synthesis and assembly inside of cells. Okay, so it's not like don't think of it like a receptor that is on the outside of a cell and then finally binds with a free-floating antigen. Cells need to process these antigens, these long peptides inside the cell, digest them, and then produce a combination of MHC and peptide at a given time. And MHC complexes are not stable without that peptide. And in the last part of this chapter, we'll talk about that actual processing, but it's in part determines what class molecule gets expressed with a particular peptide. So for instance, class 1 molecules acquire their peptides from digested cytosolic proteins, whereas class 2, I want you to start thinking phagocytes associated with class 2, like macrophages, acquire peptides from extracellular proteins and ingest them via endocytic vesicles. After the synthesis and delivery to the cell membrane, an MHC peptide association is a saturable interaction that is a very slow off rate compared to any receptor ligand binding on the order of hours to days. And this helps ensure and maximize the time that an antigen could potentially be recognized by a T lymphocyte. If that were to happen, only a very small number of peptide MHC complexes are needed to activate that specific T cell. It's actually only about 100 complexes of a particular peptide that's needed in order for that to happen. Now, I like this particular fact for 2 reasons. One, it highlights how few of these complexes are necessary to interact with the T cell in order to activate it, right? So 100 on cellular terms is just insanely small. On the other hand, it starts breaking down the notion that there's one receptor, one antigen that might kick off this magical activation process. So whenever we look at these figures in the book that are somewhat oversimplified, we have one antigen presenting to one T cell that only has one T cell receptor sticking out, and we kind of think, oh, only one is needed. But again, this kind of highlights the need to cluster many of these MHC peptide molecules, to cluster many T cell receptors. And it's the same thing with B cells. It's the same way with activating cells of the innate immune system. It's never just one receptor. It's a combination and clusters of many receptors that are always required at a given time. So don't let those cartoony drawings, as great as they are, throw you off on that fact. And lastly, I also want to dispel the notion that MHC molecules only process foreign antigens, pathogens, microbes. They're also capable of binding and processing self-antigens and peptides. In fact, most of the peptides that are displayed by any antigen-presenting cell are derived from self-proteins. And this is part because these cells don't understand what exactly they're processing at any given time. They take a protein, they break it down, they express it. And most of the proteins in the body, of course, are made by ourselves. So that's why most MHCs are presenting self-antigens as opposed to foreign ones. And it's only the T cells that recognize what's foreign, what's self. And this is a good way for T cells that are specific to self-antigens to get eliminated this way, right? So usually there's a lack of an innate immune response. So if they recognize something without other indications, activators of something that's foreign or dangerous, then that'll clue them in that they are recognizing self-antigens, and then they'll get eliminated. Now, the antigen receptors of T cells recognize both the antigen peptide and the MHC molecule. Of those two, it's the peptide that gives the T cells their fine specificity, and then it's the MHC molecule that gives them their class restriction. Now, being that antigens are typically derived from very large proteins and not short peptides, cells must first undergo antigen processing to convert these large protein antigens either in the cytosol or internalized from the extracellular space into peptides suitable for binding. Now, because this is actually where MHCs also get processed and made, this helps ensure that the MHCs are in the same cellular location as the antigens. It's also important to note that this occurs before the cell surface expression of the MHC. So again, MHCs aren't just free on the surface of the membrane and then bind to free-floating peptides. They have to get made internally within the cell along with how the antigen is processed, and then they get assembled at the same time. And because of this, the actual processing of the antigen is in itself an integral component of the biosynthesis and assembly of the MHC. Now, there are two key differences in the assembly and processing of the class 1 and class 2 MHC molecules. First is that cytosol proteins are degraded by proteasomes in the cytosol specific for class 1 MHC molecules. On the other hand, extracellular proteins ingested by phagocytes are degraded by lysosomes to generate peptides for class 2 MHC. So I want you to start thinking phagocytes, class 2, class 1, any virus or whatever that can get into the cytosol and not endosome is designated for class 1 MHC molecules. So now let's talk about antigen processing for class 1 MHCs in a little bit more detail. So there's a common association between viruses and class 1 MHCs because all viruses encode proteins that are synthesized in infected cell cytoplasm. This means that it's guaranteed that if you're infected by a virus, a class 1 MHC will somehow be involved. Now, there are other pathogens and bacteria that are associated with the cytosol region, typically through one of two ways. It could be extracellular bacteria, inject proteins into the cytosol directly. It could also be phagocytosed organisms or bacteria whose proteins are transported from the endosome vesicle into the cytosol. And this is actually how viruses actually commonly infect cells too, is that they escape endocytosed vesicles into the cytosol. Now, the first step in processing these large protein antigens is to encounter proteasomes. Proteasomes are housekeeping enzymes found in the cytoplasm of nuclei of most cells, whose main job is to degrade damaged or improperly folded proteins. Now, the error rate in protein folding is incredibly high, around 20%. So proteasomes' main job is really just to clean up things that the cells have done wrong in the first place. It's not necessarily to process pathogens or viruses in the cytoplasm. Now, while proteasomes are around at all times just for these general housekeeping functions, their expression is increased by the cytokine IFN gamma, which makes production of proteasomes more prone to cleave peptides suitable for class 1 MHCs. At this point, proteasomes have cleaved the large proteins into smaller peptides capable of being processed by the class 1 MHCs. But in order to do so, these antigens must be brought into the ER through a transporter that is creatively named Transporter Associated with Antigen Processing, or TAP. So I find that's very easy to remember. So at this point, TAP brings in the newly formed peptide antigens into the endoplasmic reticulum, or ER, where an empty and unstable class 1 MHC is conveniently waiting nearby due to a molecule called tapicin. So this kind of binds and links the TAP transporter with an empty MHC molecule. So it's directly in the vicinity of this transporter, which is why peptides are so easily brought to a class 1 MHC molecule. Within the ER, there's a final enzyme called ER-associated peptidase that can trim peptides to the proper length for MHC binding. Now, the binding of a peptide with an empty class 1 MHC reduces the affinity of tapicin, and this allows the class 1 MHC to unhinge from TAP and then proceed to processing to the Golgi complex of the cell. Now, all MHCs, regardless if they have a peptide antigen attached to them or not, attempt to go through this process to the Golgi apparatus, but it's only ones that are stable that efficiently pass through because they have a bound peptide. Those that fail to find a peptide are misfolded and transported to the cytosol and eliminated by proteasomal digestion. Finally, at the end of this route, stable MHC peptide combinations are transported to the surface via exocytic vesicles and are available for T-cell presentation. And what I like at the end of this picture here is that we see both an MHC peptide T cell receptor, along with the previously unseen CD8 plus docking co-receptor that's even binding to the site, the B cleft of the MHC complex. Now, onwards to Class 2 MHC antigen processing. Most Class 2 MHC-associated peptides are derived from protein antigens that are digested in the endosomes and lysosomes in APCs. And this could happen through a variety of processes like endocytosis, pinocytosis, phagocytosis, and other similar processes. In previous chapters, we learned how dendritic cells and macrophages express a variety of surface receptors, like lectins that recognize structures of many microbes, toll-like receptors. Macrophages also express receptors for the Fc portion of antibodies and complement receptors like C3b. And those are all what initiate the process of phagocytosis and the creation of endosomes, phagosomes, and lysosomes that eventually degrade those proteins. Less often, cytoplasmic and membrane proteins can be processed and displayed by Class 2 molecules. This can actually result in the enzymatic degradation of cytoplasmic contents, a process known as autophagy, where the cell actually starts digesting itself. And on this note, I'd like to congratulate Yoshinori Oshumi of Japan, who recently won the Nobel Prize in Medicine for his study and discovery of these self-eating cells. And this was just awarded 2 years ago, 2016. Now, while these protein antigens are being processed by endosomes, lysosomes, and later stage phagolysosomes, Class 2 MHCs are being built in the endoplasmic reticulum. Now, if you remember, Class 1 MHC construction is also occurring here, and you have the TAP transporter that brings in processed antigens from the cytosol to the same place. So there's a special molecule called the invariant chain, which is a protein that binds to Class 2 MHCs in part to keep them stable, but also to block the potential binding sites of peptides that were brought in by TAP. And this is what directs those proteins from the cytosol, those peptides from the cytosol, preferentially to Class 1 MHCs, because this invariant chain is blocking potential binding sites on the Class 2 MHCs. The invariant chain also makes Class 2 MHCs stable during their transport to the Golgi complex. I had previously mentioned that, like in Class 1 MHCs, that potentially wouldn't find any peptides to bind to. This is the process, the transport system that they would typically not survive and then be digested in the cytosol and recycled for future constructions of other MHC complexes. From the Golgi, Class 2 MHCs, along with their invariant chain, are transported to endosomes where they can potentially meet up with endosomal or phagosomal peptides. And it's here that the invariant chain just associates from MHC through both proteolytic enzymes and then a special molecule called HLA-DM. HLA-DM is actually encoded by the same MHC genes that create the Class 2 MHCs. So they're structured very similar, and if you look at the pictures here, they actually look very similar to each other. So in the first step, the invariant chain is partially digested by proteolytic enzymes, and while the Class 2 MHC molecule is stable to these enzymes. And the second step, HLA-DM, helps bind to the Class 2 MHCs in part to prevent low affinity peptides from binding to it. So it helps only the high affinity peptides destined for the Class 2 MHCs to actually bind to it. Once a high affinity peptide comes along, it displaces HLA-DM, and then a fully formed Class 2 MHC peptide combination are created. The stabilized MHC peptide combination is then brought to the surface for recognition of CD4 plus cells. And again, in this drawing, what I really like is that it shows both the T cell receptor, the antigen, the Class 2 MHC, and now for the first time, the CD4 co-receptor on the CD4 plus cell that recognizes this docking and co-receptor part of the beta chain of the Class 2 MHC. Now, one particularly cool feature about dendritic cells is one that I want to highlight here. It's the idea of cross-presentation, where another type of cell that could be infected by a virus or gotten cancer is recognized by a dendritic cell. So even though that one cell is displaying Class 1 MHCs, like all cells are able to, a dendritic cell will recognize this, phagocytose that infected cell or cancerous cell, and then display their antigens on their own Class 1 MHCs. Now, the reason this is important is for two reasons: that cells other than dendritic cells can get infected or get cancer, and dendritic cells are the most important cell for activating naive T cells. So it's not always enough that just any cell that's infected can display their antigens to a T cell. It might be necessary that a dendritic cell come into play, digest it, and then present that same antigen on its own class 1 MHC because it's a much more capable and professional antigen presenting cell. Now in this process, it might seem to violate the rule of phagocytosis materials get displayed on class 2, cytosolic materials get displayed on class 1. However, during the phagosomal process, the dendritic cells actually let these antigens escape to the cytosol specifically so they can process their antigens for class 1 MHCs. So that rule of where the antigens originate and what class MHC gets processed through them is never violated because of this. Now, some important takeaways from this chapter is that the location of a presented protein antigen determines the type of response from the T cells that it's achieved. So a cytosolic protein that's produced by viruses or tumors enters the class 1 MHC pathway and thus gets recognized by class 1 restricted CD8 plus cells that's necessary to kill that infected or cancerous cell. However, extracellular microbes or bacteria can't be killed directly by CD8 plus cells. And for that reason, extracellular protein antigens enter the class 2 MHC pathway and thus are recognized by class 2 restricted CD4 plus T cells. Now these cells help other cells like B cells produce antibodies or activate macrophages to sort of indirectly kill that pathogen. Also, something that wasn't really discussed in this chapter is that only a small portion or epitope/determinant of a complex protein might elicit the strongest T cell response. And we call these epitopes the immunodominant epitopes. And it's no coincidence that these epitopes are ones that bind most avidly, that is to say with the highest affinity, to these MHC molecules. So you can break up a protein into many short individual peptide sequences, but again, not coincidentally, the ones that bind strongly, most strongly to MHC molecules are the ones that are immunodominant and produce the strongest response to T cells. Using this, engineers can actually predict which one of these amino acid sequences associated with pathogens or viruses would be most likely to bind with the highest affinity to MHC molecules, either using in vitro methods or even now high-driven computer in silico methods. And we can use this to create synthetic proteins that have the same antigens that will be presented in T cells to create sophisticated vaccines without actually using the harmful virus epitopes themselves. And lastly, there are some non-peptides that can be recognized by T cells. Small molecules such as metal ions, nickel, beryllium are famous examples, can bind to self-peptides, alter their conformational shapes in ways that they wouldn't be recognized as self-antigens, but would be recognized as foreign antigens. And these are the source of many hypersensitivity disorders, which I think are discussed in chapter 19. But they make them capable of binding to MHC molecules because they now seem foreign to the body. There's also special subtypes of T lymphocytes, such as the natural killer T cell, and I hope that doesn't confuse you because it's different from the natural killer cell and then the killer T cell, which is also called the CD8 plus cell. So natural killer T cells can recognize lipids that are displayed on the, again, hopefully not confusing, but the class 1 like but non-classical MHC molecule, CD1. So that concludes this chapter on antigen processing and display of antigens to T lymphocytes. We learned a lot of cool concepts like class restriction of the various subclasses of T lymphocytes via the MHC complexes. We learned how the processing of proteins from different sources can help direct what kind of response the adaptive immune system comes back at, primarily through where these proteins are sourced and their presentation to either the CD4 plus variety or the CD8 plus variety. In the next chapters, we'll get into the function and activation of those two subtypes and how then they become effector cells and perform out their functions. So, thanks for listening.